So again, the quiz is going to have Chapter 5 on it, but the quiz is until next week. We're probably going to get through about half of it today, and maybe the second half Tuesday in the beginning, maybe all of Tuesday, and we'll do a review next Thursday before the quiz and take the quiz the second half of Thursday's class. Does that sound good to everyone?
No.
No.
The review Tuesday.
The review Tuesday?
Yeah.
Well, okay, if we can get through, we can maybe have the review the second half. No. I'm sorry. Well, but we can maybe finish Chapter 5 by the first half of Tuesday and then have the review. But I don't want to rush us. Yeah. The quiz will probably have like 12 questions, like 6 multiple choice and 6 short answers. Short answer like 1 to 2 sentences. No. Short answer, actually people usually do better.
Yeah, I can do matching.
Yeah. What's that? Review. Yeah, that's. Well, we'll see what we can get done. All right. So goals for Chapter 5. So we're going to talk about plasma membranes because I think membranes are really cool. So we're going to talk about plasma membranes, what they are and how they function. We're going to talk about diffusion and osmosis, which are how different particles are moving around in water and also through membranes. We're going to talk about active and passive transport. So we talked about the Golgi apparatus and how it's moving things around the cell. So things go in and out of the cells too, and sometimes that's active, as in it like requires energy. And for energy, we would probably use, what would we use for energy to move things in and out of the cell? ATP. Great. Yeah, so that's right. ATP is what we would use for active transport to power things in and out of the cell, or passive transport means they just naturally go without energy in and out of the cell. And we want to talk about the structure of membranes and how it relates to their function. This is a theme that we're going to keep seeing throughout the class, because I think the important thing about biology isn't just to memorize lots of, disparate facts, but to learn about patterns and how chemical structure is going to dictate the biological function. All right, so membranes. So they're surrounding cells and they surround organelles, but we're going to have membranes surrounding organelles only in eukaryotic cells and not prokaryotic cells. And again, they're going to be made up of phospholipids, like we see here. And these phospholipids are going to line up with their tails facing inward and their head facing outward towards the water. So we have a polar head group and a nonpolar tail. So this is the chemical structure of the phospholipids that make up the membranes that surround cells and organelles. So the function of the plasma membrane is they're going to compartmentalize cells and the individual organelles. So we talked about lysosomes, which have a very low pH and they can digest biomolecules. What do you think would happen if a lysosome had no membrane around it and it just like burst open and let everything loose? Would that be good for the cell? No, it would just break down everything in its path, and then the cell wouldn't function and it would die. So that's why we need membranes around different organelles to keep them functioning and keep the cell healthy. And also, the membranes around cells and organelles, they don't keep everything out. They let certain things through, and the cell can use those properties to its advantage to keep it healthy. Again, these are made up of phospholipids. So what type of biomolecule is a phospholipid? It's in the name, it's a hint. Lipid, great. So it's not a protein, it's not a carbohydrate, it's not a nucleic acid, but it is a lipid. We're talking a lot about biomolecules moving forward, which is why we spent some time in different regions of the cell as it relates to the plasma membrane or the stretch of phospholipids that are surrounding the cell. So outside of the cell, we have what we call the extracellular space. So extracellular means like outside of. Inside of the cell, we have the intracellular space. And that's going to be composed of cytoplasm. So cytoplasm is the fluid inside of the cell. And the cytoplasm is just water just filled with biomolecules that are necessary for the cell to survive. So we have the cytoplasm that's kept in the intracellular space by the plasma membrane, which surrounds the cell and makes it separate from the extracellular space. So if we talked about something going from in to out of the cell, we would say it went from the extracellular space into the intracellular space.
Is plasma membrane the same thing as the lipid bilayer?
Yeah, yeah. So it'd be like saying the wall versus like the bricks. So the bricks are like the phospholipids, and they make up the wall, like the plasma membrane. Okay, so just to review, make sure we're on the same page. So we have water on both sides, in the intracellular space and the extracellular space, and we have this phospholipid bilayer that keeps them separated. And again, we have the hydrophilic area outside facing the water and the hydrophobic area inside away from the water. Complicated, but I don't know why that went away. But I'm sure we can get through it. So this bilayer, this phospholipid bilayer, it's not static. It's not like a brick wall where things just stay where they're put. Things are going to move around. So the phospholipids can move side to side like this, or they can spin around in circles, but they're never going to flip end to end. So this phospholipid here will never end up over here. So they can move around side to side or rotate, but they're not going to flip end to end. And this is called the fluid mosaic model. So fluid means like not static, not staying still. I don't know not permanent. So yeah, so it'd be like dynamic, like fluid. Mosaic means it's made up of lots of different pieces. All of these different phospholipids are going to be moving around. And the way they move around is going to tell us a little bit about what materials they're going to let through and they won't let through. So within the phospholipid bilayer, we have proteins that are embedded. So the plasma membrane is not just phospholipids, we also have proteins in it. And you don't have to memorize all these, I just wanted to tell you, because we were talking a little bit about how cells know what to do. So one of the ways they know what to do is they could have these receptor proteins. So these are proteins that are going to be embedded in the phospholipid bilayer, and when they receive a certain molecule, a certain signaling molecule, they're going to change their behavior, and so they're going to send signals into the cell. So proteins can also, they can let things through either channels, or they can carry them through by using energy to do that, or they can just let them like float through freely. They can signal to other cells. So we saw earlier in the picture with the cool colors and microtubules that cells were touching each other. So cells have to communicate with each other. And one way they can do that is through what's called cell recognition proteins. But these are just like little fingerprints on the outside of the cell, so other cells know what they are. So for instance, if you get like an organ transplant, you have to make sure, or doctors will make sure that the cell recognition proteins on the outside don't make your immune system attack that organ. So it will recognize it as yourself and not as something else. Your immune system uses these cell recognition proteins frequently to know whether things belong in your body or not.
Is that related to blood type?
It is related to blood type, but it's not exactly the same, and I'm not totally sure what the difference is, to be honest. Good question, though. But yeah, I'm pretty sure that blood type just relates to what cell recognition proteins are on the outside of the blood cell. But I'm not sure how it would relate to like organ transplants.
It's all connected somehow.
Yeah. Would be, maybe I should do extra credit things, and that could be one of the topics. Let's see how the first quiz goes. If it doesn't go well for everyone, we can talk about extra credit things, and that could be one of the topics. Okay, so we talked about proteins that are in the phospholipid bilayer. So they can be in different regions of the bilayer. So they can be what we call transmembrane proteins. So this goes all the way through. So it has some of it on the outside of the bilayer and some of it on the inside. We also have peripheral proteins. So they'll just be sticking out on the outside or the inside. All right, sorry, only intracellular space. I'm not sure that's true. I think I might have made a mistake. The important thing to remember, though, is that they won't be facing both sides, not extracellular and intracellular. So we have peripheral and transmembrane. So what do you think would cause this difference in function? Why are some proteins going to be spanning the entire part of the plasma membrane, and some just on one side.
Some have non-polar.
So, what?
Some have non-polar molecules, so they're not spanning.
Good. So, what? Maybe elaborate a little more. Some proteins have nonpolar.
They might have non-polar molecules attached on one end.
Yeah. So, where would the nonpolar regions of a protein want to be?
On the inside.
On the inside of in the nonpolar area. And where would the polar regions of the protein want to be?
All the way through.
Yeah, either here or here. So, exactly. But everything's online. I put it all online, so. Okay.
It's okay. Go ahead.
Well, I'll try to go slower. So this is a representation of a protein, and so if we look at each amino acid one by one, we get its primary sequence, and this tells us a little bit about its tertiary sequence, its 3D shape. So these green amino acids, these are nonpolar amino acids, and the nonpolar amino acids are going to want to be right here, which is going to be in the plasma membrane. And the polar ones are going to want to be mainly on the outside or the inside. So it's not, totally perfect. It's not that all of the nonpolar are on the inside, all of the polar on the outside. But generally speaking, we have the nonpolar ones on the inside and the polar ones on the outside. So based on the instructions that the ribosome gets from the DNA, which is in the nucleus, it's going to produce a very specific sequence, the primary structure of the polypeptide chain of the protein that's going to have either polar or nonpolar proteins, or amino acids, sorry. And that's going to dictate where the protein ends up in the plasma membrane. So if we wanted a protein in the plasma membrane that would be a transmembrane protein, we would have to have some of these nonpolar amino acids. And based on which nonpolar amino acids we have, it's going to dictate which parts of the protein are inside or outside or in the plasma membrane. So this is how the structure of a protein is going to dictate its function in relation to where it is in the plasma membrane and what is seen outside or inside of the cell.
So in the previous slide, the picture, the protein was like this blob sticking through the cell.
In addition to having proteins, we also have carbohydrates. And these carbohydrates are like little flags or ID tags. They're kind of like the fingerprint of the cell. So it's important for cells to be identified as part of your body, because part of what your body does with your immune system is it makes sure that nothing that isn't there or that shouldn't be there isn't there. And if it's not where, if it's foreign, if it doesn't belong there, it attacks it and tries to digest it. So if your immune system bumps into one of your cells, it's going to check its glycolipids or glycoproteins to see if it belongs in your cell, to check its fingerprint and make sure it belongs there. And so what glycolipids are these are lipids that have a carbohydrate chain attached to them. So a carbohydrate chain, is that going to be a monosaccharide, or what do you think that would be? Polysaccharide. So there'll be a chain of many monosaccharides. A glycoprotein is a carbohydrate attached to a protein. So again, this is just kind of give you some idea that the plasma membrane isn't just some, permanent wall around the cell. It moves around, it lets some things through, it has proteins in it, it has carbohydrates in it, interacts with other cells, it does a lot of, useful functions.
So do the glycolipid protein have the same function? Or
at this level, yeah, you could say they have the same function, but if you dug more into it, they have.
But it's just like
Exactly, yeah.
What do you mean by combination?
So when your immune system would bounce into a cell, it would check a number of different glycolipids and glycoproteins to make sure they all match.
Well, depending on whether or not it's a lipid or a protein that change, like bonds and whatnot, that are kind of like the polar versus non-polar.
Yeah. So, sorry. What do you mean? Sorry. Could you explain
So
the
like that's how they would interact differently. Like the lipid specializes in a certain polar or non-polar kind of interaction. The outside of the protein interacts with the opposite.
Yeah, I think maybe if we think about it kind of as if these are going to be made in your endoplasmic reticulum, which is like sort of like a workshop, you might say, you'd be attaching carbohydrates to either the lipids or to the proteins, and you would attach it to different molecules based on where you wanted it to go, and you would have a different sequence of these polysaccharides to give it different like signaling capabilities. Does that sort of answer your question, or is that not?
Yeah.
All right, cool. All right. So that's just, we can take a few, do you want a few minutes to go over these, or do you just want to answer them as a class? Answer them as a class. You guys don't like talking to each other? That's fine. How do phospholipids form membranes? Why don't you raise your hand if you have an answer? Maybe someone who hasn't spoken yet would be nice. I could call on people. That's not fun. You guys already answered though. Is anyone like in this? Yeah. What?
Figure out phospholipids form a phospholipid bilayer, which forms the plasma membrane.
Great, exactly. So phospholipids have a polar and nonpolar region. We put the nonpolar region inside, and we have a bilayer. So we have two layers with both of their tails inside and their heads outside. Great.
So you said it forms a bilayer that forms the membrane.
Exactly.
Okay.
Yeah. So are these membranes, are they static or fluid?
They're both fluid.
Or someone are, yeah, Chris. Fluid. And so how are they fluid? In what ways do they move around?
So they can circulate.
Yeah, great.
Moving back and forth.
Exactly. Great.
Within space.
Yeah, but they're not going to move. So if this is, if we have our bilayer kind of going this direction and these are phospholipids, they're not going to ever flip around like this. And the reason they wouldn't do that is because you have like a polar head here, and that really doesn't want to be in the nonpolar space. So it'll just move in this direction and it'll spin around, but it won't flip over. And what's the model called to describe how membranes are moving around?
What's that?
Fluid mosaic model. Great. This is fun, isn't it?
Yeah.
Great. We're all enjoying yourselves. So what are some other components of membranes and what are their roles? Does anyone name one of them? What's one part? Glycolipids. Great. So what else is similar to glycolipids? Glycoproteins. And what do they do?
You just hang there.
They're just hanging out. And what do they? Does someone want to raise their hand who hasn't answered? Anyone who hasn't answered? Trying to spread the communication. It's fun. Start talking. I have to talk the whole time, so you can at least share my commiserate with me.
Attached to them.
Yeah, but what do they do? How do they interact with other cells? What do they do with other cells? Do they destroy them? Yeah, it's like a combination. It's like a key. It's like a fingerprint. It just lets it know that it belongs there. So if you have some bacteria that gets into your body, it's not going to have the right glycolipids and glycoproteins, and your immune system is going to see it eventually, and it's going to realize it doesn't belong there, and it's going to try to destroy it, and hopefully it will. Otherwise, you could get an infection.
Can't some bacteria and viruses like copy these?
Yeah. So it would be great if I had an example of this off my head, but I imagine there's some bacteria or some diseases that can replicate the glycolipids and glycoproteins of your own cells to make it look like they belong there, but in reality they don't.
Well, in high school we learned about HIV and AIDS, and it kind of sounds like that's.
Yeah. I don't know. It might be. Yeah. Yeah, it could be. I'm not sure.
There was a video where like the HIV virus was basically dressing up like the white blood cells. So
Oh, okay.
it sounded like maybe they were mimicking the fingerprints.
Right. Yeah, and that way they could travel more freely around the body without getting destroyed. So we talked about glycolipids and glycoproteins. Is there another biomolecule that hangs out in the plasma membrane? Proteins, yeah. And proteins can have a wide variety of ranges in the plasma membrane. Sorry, a wide variety of functions in the plasma membrane. I forget what I had said. All right. So one of the things that's important with membranes is what they let through and don't let through. And so we're going to start off talking about passive transport. So passive transport is transport that doesn't involve energy. You don't have to spend any energy to make this happen. It just happens by itself. So one example of passive transport is diffusion. So diffusion is the movement of substances from an area of high concentration to low concentration. And we all kind of have an intuitive idea of what diffusion is. Like if you put some sugar in your sweet tea in your drink, that sugar is going to diffuse throughout the entire tea until it all has the same concentration. Sort of like this excellently drawn diagram. If you put in some solute is like the substance that you're adding. So if we were doing the Kool-Aid lab, it'd be like the Kool-Aid that you add to the water, which some of you will do for the first time today. Maybe not the best example. But if you add Kool-Aid to water, the Kool-Aid is like the solute and the water is like the solvent. No, we did that in lab 1, so everyone should understand this example. Great. So when you add the solute, if you just let it sit there for long enough, it would completely diffuse throughout your liquid. That's diffusion. So it would go from areas of high concentration, like this is an area of very high solute concentration. It would diffuse to areas of lower concentration. So now it's spread out to areas that previously didn't have any Kool-Aid. This is diffusion. So the movement of stuff from areas of high concentration to areas of low concentration. Does that make sense? This just happens naturally. You don't have to add any energy, but if you do add energy, it goes faster, but you don't have to.
So you
It's
don't
passive.
have to have energy to do a passive train.
Exactly. Great. So we also get diffusion through plasma membranes. So if we have a bunch of stuff outside in extracellular space, and this stuff can pass through, it's not too big, or it doesn't, for other reasons that we'll discuss later, not fit through. If it's possible that it can go through, over time, it will eventually diffuse through the plasma membrane. So it goes from an area of high concentration to an area of low concentration. So this is how your cell can get some nutrients, just by diffusion. Just by things that are in your blood, and your blood perfuses all your cells, it can get into your cells. So like oxygen is a good example of this. We have oxygen in our blood, but we need oxygen in our cells. And luckily, oxygen can pass through the plasma membrane because it's so small and it's not charged, which we'll talk about later. So oxygen can diffuse from an area of really high concentration in your blood to areas of low concentration in your cells. This is the way that your cells can get nutrients. So luckily, our plasma membranes aren't like actual walls that are totally impermeable that nothing can go through. Otherwise, you wouldn't get any oxygen into your cell. This is an example of passive transport through a membrane. So not everything diffuses through. So small things that are uncharged, like water, can pass through. So small things that are hydrophobic. So O2 is hydrophobic. So why would O2 be hydrophobic? Any guesses on that? What does hydrophobic mean again?
It's
Yeah, or it's polar or nonpolar? Nonpolar. So why would O2 be nonpolar? Someone raise their hand. What?
It doesn't have the hydrogen. Light.
That's moving in the right direction. You're on the right path. Does anyone else have a?
The O2 is it has the same electronegativity with the bond of oxygen and oxygen.
Great, exactly. So what makes something polar is when there's a difference in electronegativity. And since it's two oxygen atoms bonded together, they have the same electronegativity, so this is going to be a nonpolar covalent bond. And so since O2 is nonpolar, it can pass right through. Same with nitrogen. CO2 is actually also nonpolar, which is a little bit counterintuitive since we know carbon and oxygen have different electronegativities. But luckily, they're different in very symmetrical ways, and so it actually ends up being an equal sharing of electrons because you have two oxygen atoms on each end of the carbon. They're both pulling with the same strength, and so they actually both end up sharing it equally. It's like if you're playing tug of war and there's just like a little kid standing in the middle holding the rope. Technically, the kid is weakest, but since you and the other person are the same strength, everything kind of stays equally shared. So water is small and polar, and it can go through. But big things like glucose and sucrose, these are carbohydrates. Glucose is a carbohydrate that we need to survive. It doesn't just pass right through because it's too big. And then things that are charged or called ions, they don't pass through either. So small things and non-polar things can diffuse through the plasma membrane, but big things and charged things can't. So how do you think we get glucose into our body if it doesn't, or into our cells if it doesn't just diffuse through the plasma membrane? Any guesses? That's how we get into our body, but how do we get it into our cells?
We actively open it.
Enzymes break it down.
No, that's a good guess, but what anyone, maybe someone who hasn't answered yet. Talking in class is really fun, I promise you. How does glucose get into our cells if it can't diffuse through the plasma membrane? So I'll leave you in suspense, and we'll cover it very shortly, I promise. All right, fine, we'll talk about it now. We have to use ATP to get it in. We have to use energy. So that would be active transport. I can't keep secrets. I'm not good at it. All right, so we know that water can go through plasma membranes, but not everything else can. So when we say membranes let some things through, but not everything, we say those are selectively permeable, or we could say semi-permeable. So they're permeable to some things, but not everything. So if you have like this example, so this is an area of high concentration, this is an area of low concentration. And so ideally what would happen is these big red balls, these could be like glucose, would pass through, they would diffuse through, and it would make the concentrations equal. But since that can't happen, what happens instead is the water molecules move out from the dilute region, the low concentration region, into the high concentration region. And that'll make both concentrations equal. Does that make sense? It takes a little bit of time.
So is that why when you eat salt or something actually dehydrates you because the water like leaves the cells? It's like
Exactly. Yeah. So I think what would happen maybe is you end up with a salt in your blood potentially, and then water has to leave your cells to make the concentration lower.
Is that why when you eat a lot of salt and then they say however much amount of salt you eat, you drink that much amount of water?
Right, to counteract the dehydration. It's making you thirsty thinking about it. So osmosis is a type of diffusion. Sorry. Hopefully it's just not me speaking. It gives you a headache. So osmosis is the movement of water through a semi-permeable membrane to make two regions have equal concentration. So water will move from an area of low concentration to an area of high concentration to make the concentrations equal.
So it is diffusion called osmosis because why is it called osmosis?
Because it's diffusion of water through a semi-permeable membrane. Yeah. Great. So that what we call the process that pushes the water to the area of high concentration, that's called osmotic pressure. That's like the pressure that pushes it through.
So it's active?
No, so it doesn't require any energy. This is also passive transport. Good question. All right, this will be our last slide. So when we talk about how cells behave in different solutions, we talk about either isotonic, hypertonic, or hypotonic solutions. So if something is isotonic, that means the concentration inside and out of the cell is the same. So is there going to be any diffusion of water here by osmosis? No, because these areas have equal concentration, so you don't need to do anything to keep them equal. This is hypertonic. So this means the solution is hypertonic. That means the solution has a higher concentration than the concentration inside of the cells. So which way would water go in this situation? Out of the cell or into the cell?
water would go out of the cell.
Oh, like through the cell. I thought that's what you mean.
Yeah. So if you have water in the cell and outside of the cell, which way is water going to move? Out. So water's going to move out of the cell because that'll make this concentration lower. As you add water to this, it's like the concentration goes down. Does that make sense to everyone? If you have a question, that would be.
I thought you were talking about like that movement of action.
Okay,
Yeah, it's hard to get their perspectives right.
Which way are you starting from, inside the cell or outside?
Well, just like the net movement of
So you said
water.
outside.
Yeah, water will move out of the cell more than it will move into the cell. So this would be a hypotonic solution. That means the solution has a lower concentration out of the cell than inside of the cell. So which way is the water going to move in this situation? Into the cell. So if you had a cell and you put it in pure water, which way would you think the water would move? Into the cell. And what do you think would happen if too much water flowed into the cell? It would burst and it would die. So if you have cells, if you're in a research lab and you have to work with a bunch of cells, you can never put them in pure water. You have to put them in like a saline solution to keep the osmotic pressure low, stop them from bursting.
Is it a word for that? I'm not red line.
For what?
Like if it
Bursts.
burst.
It's called lysing. The cell would lyse. Maybe there's another word. All right, I think this is a good place to stop today. I think we can probably finish this on Tuesday and then review the rest of the class on Tuesday. That'll be the plan.